We're up to our 10th lecture now, and this is covering chapter 6. We're going to be discussing enzymes. And a lot of these slides are from a previous class that I taught, where I had to take over for someone that got sick at the very end of the quarter at the University of Denver. So I just posted some lectures, online lectures, that I thought covered the material we were going to be covering the rest of that quarter pretty well, and so they're included here. You're welcome to look at these if you think it would help. They covered enzymes that we'll be talking about today, and they also go into kinetics, some mechanisms, and some regulation. So we'll be covering all of this in maybe more or less detail. So the main thing is to stick with the book and the homework problems for sure. But these are some extra lectures. If you don't find the lectures that I'm giving very helpful, or if you just want something extra. And what are enzymes? The word enzyme really means a catalyst. Okay, and remember what that is. A catalyst is something that increases the rate of a chemical reaction without being used up itself in that reaction. Okay. Most enzymes are actually globular proteins, and that's another thing I should mention about enzyme. An enzyme is a protein that is a catalyst. Okay, a protein that catalyzes a reaction. Okay, most are globular proteins. However, well, actually what I just said is a little bit untrue, because some RNA are actually able to catalyze reactions as well, which you would sort of consider an enzyme, but really they're a little bit of their own thing, ribozymes. Okay. This is really one of the study of enzymes is really probably the earliest work in biochemistry was done studying enzymes, okay? Dating back to the late 1700s. Okay, and it was probably not surprising, because people would, trying to make food or wine, they would have these biological extracts, and they would see that they're creating something different, like with yeast fermenting, or things like that, and then they'd want to study what inside this is making that process happen. Okay, and it really has dominated the study of biochemistry in the past, and it will continue to do so. It will always be a big part of biochemistry, at least for the foreseeable future. Okay, so why do the cells use biocatalysts over inorganic catalysts? Okay. So these are three pretty important reasons why enzymes are used instead of inorganic catalysts. Okay, one, biocatalysts or enzymes give greater reaction specificity. Okay. So they have more specificity in the reaction, and they avoid side products. And so side products in a reaction might be very detrimental inside a cell. Some of the side products could be toxic, so it's good to avoid those. Okay. You can use milder reaction conditions with enzymes, so that's conducive to conditions inside of a cell. Higher rates of reaction, and that's really, probably any catalyst you use would give, whether it's biocatalyst or inorganic catalyst, it's going to give you a higher rate of reaction. And that's so you could get these reactions done in a biologically useful time frame. And then finally, a very important one with enzymes is they have a capacity for regulation. So you can control these biological pathways very well. And the figure down here is just sort of explaining this. If you have a reaction for this molecule, right, and you want to change it into this molecule, there's other reactions that could occur, but this enzyme catalyzes this one reaction very specifically, and it makes the desired product, and you don't get the side products. So this is a figure that really explains the idea of a reaction occurring on a biological useful time frame. Okay. So we're plotting the amount of product formed over time. Okay. And you see that in black here, this is the uncatalyzed reaction, and it goes, but slowly, and it's sort of linear, okay? And it would take hours to reach a high product amount, product concentration. But that reaction, that same reaction catalyzed by an enzyme, the product concentration is going to increase very rapidly, very quickly. So in the order of seconds, you can get a very high product concentration, whereas when you don't have a catalyst, no enzyme, it would take hours. And this table really illustrates that fact. Okay, so some of these we might see again, especially next semester in metabolism, triose phosphate isomerase. And we've talked about carbonic anhydrase a little bit, made mention of that one. These other ones, not so much. But it's still interesting to look at what the half-life of some of these reactions are without the enzyme and with. So this enzyme here, OMP decarboxylase, which I'm not really familiar with, but they give that half-life of that non-catalyzed reaction as 78 million years. Okay, crazy, right? That's super long for a half-life. Okay, so the uncatalyzed rate is given here. 10 to the minus 16th, very small rate. Catalyzed rate is 39 per second. Okay, not a very large number, but compared to the uncatalyzed rate, huge enhancement. So the enhancement is 1.4 times 10 to the 17th. That's an enormous factor of difference, of increase, right? So you can see that enzyme speeds that reaction up so much, right? And some of these other ones are kind of interesting to look at too. There's ketosteroid isomerase, right? The catalyzed rate is 66,000 per second. That's much bigger than 39, obviously, but that's a huge number. Okay, so some of these enzymes can really put out a lot of product. None of these numbers, I should mention that you don't have to memorize these. Okay, it's just really kind of drives home the point that enzymes speed up reactions, and they do it very well. Okay. When the substrate binds to the active site, it binds via a combination of intermolecular forces, and we form what's known as a enzyme substrate complex, and that's abbreviated as capital ES. Okay, E for enzyme and S for substrate. Okay. Once that complex is formed, the conversion of the substrate to the product then takes place. So typically you see just a generic reaction written for an enzyme as such, E plus S, and then there's this equilibrium between the two with the ES, enzyme substrate complex. Okay, that is a reversible process, so that substrate can bind and it can come out. But then there's also a second step to the whole thing where the enzyme substrate process goes to the substrate is then converted to a product. Okay, and even sometimes you see this written as enzyme substrate goes to EP, enzyme product, and then you get enzyme plus product dissociation. So sometimes you see this written as even an extra step. And then a specific example is shown down here with the sucrase enzyme. Sucrose binds to it, and you get the sucrase-sucrose complex. So sucrase is the enzyme, sucrose is the sugar, right? And then you get the product release. So sucrase is the enzyme plus the two products, glucose and fructose. And again, as I mentioned, this enzyme substrate complex is held together, is formed by weak intermolecular forces. And remember when we've talked about those before, what are those? Hydrogen bonding is one of them, salt bridges or electrostatic forces, and then simple hydrophobic interactions between the substrate and the enzyme binding site also play a role. And this is just a reminder. We've seen this slide before, right? What are the intermolecular forces? Okay, dipole-dipole interactions, dispersion forces, hydrophobic force. Those are sort of generally referred to as van der Waals forces. Okay, and then the ion-ion interactions, which would be the salt bridge, are actually, one of the stronger intermolecular forces that also play a role in certain enzyme substrate binding complexes. And I believe I've shown this before we talked about enzymes, but this is an example of an enzyme binding site. Okay. So if we start here at picture 1, okay, that's the enzyme with nothing in the binding site. Okay. Then the substrate will bind. Okay, it fits very well into a certain pocket there. But you'll also notice there's this other shape here that doesn't really fit that substrate. What that fits down here is the transition state. Okay. And the transition state also is, in this case, these hydrogen bonds here are stabilizing that transition state. Okay. So maybe this part of the molecule, the substrate molecule, fits in this pocket via, a hydrophobic interaction or some hydrogen bondings there. There'll also be some stabilization to the transition state via something like hydrogen bonds. Okay. That transition state is stabilized, then the reaction occurs, and you get your two products out. That's really, we'll be discussing this in a little bit more detail throughout the lecture. Okay, the book shows this example. Uracil is the substrate in this enzyme, and you can see there's certain hydrogen bonding interactions there. Okay, there's a serine forming a hydrogen bond, threonine side chain, and then there's actually a hydrogen bond to the backbone nitrogen amine group of that threonine. Right. And the colored amino acid numbers here are in the structure are the ones that are important that make up this active site pocket in this enzyme. Okay, and you'll notice that they're not always in close primary structure and in close sequenced with one another, but when the enzyme folds, they can become close together. right? Specificity and selectivity. Okay. This is a phenylalanine, and the example here is phenylalanine hydroxylase enzyme. Right? So when phenylalanine binds, okay, this enzyme will add a hydroxyl group to it and turn it into a tyrosine. Okay. Um, so it gets hydroxylated. Right? Now, if the other isomer of this, so this is, let's call this L, this is the D, right? If the D form of phenylalanine is added to this enzyme, there's no binding. Okay, so it's very specific to one type. Right? If something else, very similar, and I'm not sure if this might be an inhibitor of this enzyme, I'm not sure, but if this molecule that's fairly similar binds to it, it can bind in that active site, but no reaction takes place. So that's an example of the specificity of enzymes. An example of why enzymes are important biologically, being able to catalyze reactions in more gentle conditions, right? If you think about what conditions are inside of your cells right now, well, you're approximately 37 degrees Celsius, and your pH is roughly 7. Okay, those are very gentle conditions. Okay, and those conditions don't always occur. Reactions don't always occur very, very fast in those conditions. Oftentimes you'll have to, if you're just trying to, an industrial process to make some sort of a product chemically, you're going to have to use really high temperatures, really high pressures, or very acidic conditions, very basic conditions, conditions that really aren't compatible with life, but enzymes are able to do this in those conditions. Okay. So what is the source of the energy for dramatically lowering of activation energies? And that's we're kind of skipping ahead here, but what enzymes do is that they lower the activation energies. We'll talk about this more specifically here in a second. But the source of that energy is due to the fact that enzymes have very specific pockets for very specific substrates, and in those pockets you have these weak intermolecular forces, right? The ones that we've talked about. Okay, that's the source of this energy that helps to lower activation energies and make reactions occur faster. Okay. So these complementary shapes of the binding pockets in enzymes. This example here, dihydrofolate reductase, okay, in these pockets, it has two sites. Okay, it has the red molecule here is NADP. We'll talk about that. It's a very common cofactor. And then yellow is tetrahydrofolate. Okay, and both of them have very specific pockets that they fit in this enzyme, right? And you see the especially NADP, it's a very large, complicated molecule, so that it's not really, I guess, an accident that it can fit very well in that site for it. Okay. And that brings up two models when we talk about substrate enzyme binding. There's two models that are usually discussed for this. One is the lock and key model, and that's where you have a binding site that is complementary to the substrate, where only the substrate can fit in. Okay, very, very simple model. I will say that this model is probably not 100% accurate. Okay, we'll see another one here in a second that's probably a better description of reality. Okay, but this is a very simple one, a very easy one to understand. You have a substrate, it fits into that active site, right, nothing else can fit. Okay, it turns out the key does not perfectly fit into the lock. Right? Enzyme active sites are complementary, not to the substrates. In I should mention that's a little bit misleading, right? They're complementary to part of the substrate, or at least some of the substrate, but the active sites are really complementary to the transition state. And the transition state is the intermediate that the substrate pass through as they are converted into products during this reaction. Okay, so the enzyme really is complementary to the transition state. And this is where we get a more accurate model called the induced fit model. Right, so the substrate has a particular shape, and in this case, it probably, A matches up with this part and B kind of matches there. So those might, sort of bind first in the substrate, but then there's a sort of change, right? And sometimes it's the enzyme physically moving. Other times it's just the substrate changing and turning into a transition state that has that changes the structure and induces the fit. So this is the induced fit model is really a better description of enzyme substrate binding, right? And what in the shortened version of it's that the enzyme is stabilizing the transition state. Okay, not just the substrate, but the specifically the transition state. Yeah. So how do enzymes speed up reaction? Well, they don't, and this is true of all catalysts. Okay, they don't affect equilibrium. They don't affect the thermodynamics. They don't affect the delta G, the overall thermodynamics, or the delta G of the reaction. Okay, so if you have a reaction, you have your products have a certain free energy, your reactants have a certain free energy. The enzyme doesn't change any of that. Okay, what the enzymes do to speed up the reactions is that they lower the activation energy or activation barrier, or the delta G of the transition state. And so this little symbol here, this double cross, that symbol means transition state. Okay. And a transition state is something it's not, the molecule is never physically, you can't isolate the molecule in the transition state. Okay, it's just a very quick intermediate that the molecule passes through in the reaction. Okay, but the enzymes lower that energy, okay, via the intermolecular forces we talked about. The induced fit model is complementary to the transition state. Okay, and you're getting some things like hydrogen bonding to that transition state, and that lowers this activation energy of that transition state. Okay. And they increase the reaction rates by decreasing this delta G of the transition state. Okay. And so the rate equation shown here, okay, this is constant, that's the temperature. Okay, to be honest, I don't remember what H is off the top of my head. But you see there's an exponent, and you have the exponent e to the delta G of the transition state over RT, right? So changing that transition state energy is going to increase your reaction rate. Graphically, what that looks like, if we plotted, this is a reaction coordinate diagram, if we plotted free energy versus the progress of the reaction. Okay, if we have substrates here and products here, it will pass through a transition state. Okay, the uncatalyzed reaction, this activation energy is quite large. Okay, this represents the delta G of the transition state going from substrate to product. Okay, very large. Okay, if we go in the reverse direction, that transition state energy is even larger. Okay. Now, when we have a enzyme catalyzed reaction, this changes to the blue shape. Okay, and sometimes you see this with the double dips, and one minimum here would be the enzyme substrate complex, and then the next would be the enzyme product. Okay, other times you see this as just one single hump. Okay, this one's probably a little bit more accurate because it's showing you these complexes, but okay, if you see it as just one hump, that it's telling you the same thing, just not going into these complexes. Okay, but the main point is you're lowering this transition state energy. So if you see the difference between the substrate delta G and the transition state delta G, it's much lower than in the uncatalyzed reaction. Okay, so this is the delta G transition state of the catalyzed reaction and uncatalyzed. Okay. Okay, so how do enzymes lower the delta G? Well, we've sort of already talked about this. Okay, they use these intermolecular forces, but another way that this occurs is that they organize reactive groups into very close proximity and into the proper orientation. An uncatalyzed bimolecular reaction, so two molecules reacting together. Okay, what if we looked at that uncatalyzed? You have two free reactant molecules. Okay, they're able to rotate and move around in three-dimensional space. During that reaction, they have to come together and be ordered into a single restricted transition state. Okay, so that you're taking two free things spinning around and you're restricting them in the process of the reaction, restricting them into a very tight ordered transition state. That's not favorable entropically. Okay. And even in a unimolecular reaction, so one molecule, that reactant is going to be pretty flexible, but as it passes through the transition state, it becomes very rigid. Okay, that again is not favorable. So these catalyzed reactions on the enzyme, they're taking these reactant molecules and they're placing them in very specific orientations and at a very specific distances from each other so that they mimic what the transition state geometry and proximity is. Okay, and so that's how they lower that entropy cost. That you still might say, well, your molecules are free and they bind to the enzyme, and that is a entropically unfavorable, and that's correct. So the entropy cost is paid during this binding, but that entropy loss is offset by the intermolecular forces that are binding that substrate to that enzyme. Okay. Right. So if we look at this is an example of an experiment that was done to prove this point. Okay, if you have a bimolecular reaction with these two molecules free in solution, okay, they form this product. Okay, they connect together here to form this product. Okay, what you're getting, we just say that rate is 1. Okay, relative. Okay, now when we have, instead of these two molecules, we make this a unimolecular reaction. So they have the same functional groups, but now they're connected here. When that reaction occurs and they form that bridge, that ether bridge, that rate enhancement now is 10 to the 5th. Okay, pretty big. And that's showing you that just taking these two molecules and rigidly locking their orientation to one another is going to speed this up. And that's sort of what an enzyme does. It would take, the binding site would put these two very close to one another and be able to speed up that reaction. And then even greater example of that is this molecule, right? You still have some freedom of motion in these three bonds here, but in this molecule, very, very rigid, and you get an enhancement of 10 to the 8, even greater. Okay. All right. So this is not an example of an enzyme catalyzed reaction, but it's an experiment that was done to prove that enzymes can speed up reactions by locking these molecules into very close proximity and a very locking them into a geometry that's very conducive to the product being formed. So how to lower delta G transition state? Again, enzymes are going to bind the transition state the best. And this idea was actually first brought up by Linus Pauling in 1946. Okay. Enzyme active sites, are complementary to the transition state of the reaction. Okay. That means the enzyme is binding the transition state better than it binds the substrate itself. Okay. So you get stronger interaction, which means additional intermolecular interactions with the transition state compared to the substrate. And this is what lowers that activation energy barrier. Okay, this is largely a enthalpy effect. Okay, that's in that enthalpy effect, positive enthalpy effect, is what's offsetting the negative entropy effect for the substrate's binding to the enzyme. And the book likes to give this example of a generic enzyme we'll call stickase. Okay, and stickase is going to catalyze the breaking of a stick. Right, so we have our stick, which is our substrate, metal stick. Okay, the transition state is a bent stick, and the products are this broken stick. Okay, so that's the uncatalyzed no enzyme reaction, and we get this free energy curve. Okay. If we had an enzyme that was complementary to the substrate, right? So this is our enzyme in blue, and you can think of this substrate binding active site here having magnets in it that will interact with that metal stick. Okay, if that was the case, if that active site was complementary to the substrate, how would that change the free energy curve? Well, we're going to lower that enzyme substrate free energy because now there's these magnets interacting with it. Okay, but it's not doing anything to that energy of the transition state. So we'd actually get a minimum here, and then the same activation energy for the transition state. That actually makes that hill to overcome even larger than it was before. Okay, so what that would do if enzymes were really like that, substrate would bind, and then nothing would happen. Okay, in a sense, that would slow down the reaction. Okay. Enzymes that are complementary to the transition state, which is the model that we use now, that being actually the case, right? The substrate will bind in that active site. Okay, the active site fits it very well. There are some interactions with that substrate. Okay, but it's not completely complementary to the substrate. Okay. Now when we see the transition state, the bent transition state, that enzyme fits that transition state very well. It's complementary to the transition state. Okay. In the free energy curve, what we'd see with this is a dramatic lowering of that transition state energy, Okay, and that would that lowering of the transition state free energy, or what we call the activation energy, that's what speeds up the reaction. Okay, so this stickase is a very easy to understand, I think, example, and this is just another picture of that. So we'll talk about the biochemical reaction types. Okay, and this is a very easy way to characterize and categorize enzymes. There are really so far only 6 classes of enzymes that have been described. Okay, so there's thousands and thousands of enzymes in your body, but there are only really 6 types of reactions that they catalyze, and we'll go through these. Okay, this list here it's a little bit off. I probably should have changed it. I did it in this order because this is kind of the order that the book gives the examples. But if you want to be really 100% accurate with it, I should have done it in the reverse order, right? Because class 1 is oxidoreductases, class 2 is transferases, and so forth. Okay, but this was kind of the order of the book, which was reversed to what the enzyme class numbers are. And I will say that the group transfer and oxidation reduction reactions, this is more for metabolism. Those are very important in metabolism and also in signaling, cell signaling. Okay. Okay, so the enzyme classes, you'll see this EC number for enzymes, and it's four numbers. Excuse me. The first number is telling you the class of the enzyme. So what basic reaction it catalyzes. Okay, class 1 is oxidoreductases, and this is where you have a redox reaction occurring. One thing is reduced, another thing is oxidized, and that enzyme, it's transferring electrons from the reduced substrate to the oxidized substrate. And what you get out is an oxidized, so let's say A is reduced and B is oxidized in the substrate form. In the product form, A will be oxidized and B will be reduced. And you see many examples of this, especially in metabolism. Okay, transferases, as their name suggests, they're transferring something. So in this cartoon example, you have A, B, and C as substrates, and it's taking this B part of one of the substrates and moving it to C. Hydrolysis, those either add an OH group from water, or they create water. If they go in the reverse direction, they're going to take out an OH group and create a water. Okay. Basically breaking bonds using water. Lyases. Lyases are a little bit difficult to define. Typically, it's a bond breaking without oxidation reduction and without water. Okay, but then a lot of these enzymes actually do operate in the reverse direction. So instead of a bond, it'd be this way. Instead of a bond breaking, they're forming a bond. Okay. 5 is isomerases, and this is an enzyme that will convert the substrate from one isomer to another isomer. Okay. And then finally, class 6 are ligases, and ligases are also known as synthetases, and they use ATP or equivalent, GTP, something like that, to stick things together, or in the reverse case, to break them apart. Okay, so we'll go through some examples. Ligation reactions. Okay, ligation reactions, as the name suggests, you're sticking things together. Can also be breaking them apart. Okay, and ligases, again, that's class 6. Okay, if we're kind of going in reverse order here. Right, so you have two molecules and you're sticking them together. Okay. Isomerizations, you're rearranging a molecule. So in this example, phosphohexase isomerase, which you will see in glycolysis and metabolism, right? You have glucose 6 phosphate, and you're turning that into fructose 6 phosphate. So what you're doing is you're moving the carbonyl group from carbon 1 to carbon 2, changing this from an aldose, an aldehyde sugar, to a ketose, a ketone sugar. We'll talk about that, different types of sugars, when we cover carbohydrates. So don't worry too much about that right now. Okay. And the mechanisms shown here, again, probably don't need to really focus too much on this at this point in the class. This is something, when we in metabolism we get into more of the actual nitty gritty mechanisms for some of these enzymes. But what you'll notice here is that there's no change in overall oxidation state. Right. Hydrolysases, they catalyze hydrolysis reactions. Okay. And so a classic example of this is sucrase, right? We have sucrose, which consists of a glucose and a fructose monomers that are stuck together. So sucrose is a disaccharide, it has 2 sugars. So sucrase uses H2O to break this disaccharide bond and give uh, glucose and fructose monosaccharides. Okay, so that's an example of a hydrolase breaking a bond with water, but you can also do some of these enzymes catalyze the reverse reaction where you actually form a bond and water is a product, which would be a condensation reaction. So really for any enzyme class, that is, you can have examples of the reaction going in the forward direction or in a reverse direction. Uh, class, I guess this would be is this class 4? Sorry. Yes, it is. Okay. Class 4 is Lyases, and this is our fourth example. Okay. And this is where you have a breaking or forming of a bond without using water and without redox. Okay, and it's a very hard class of enzyme to really to describe. Um, but there are many examples of lyase enzymes. Okay, so if there's ever an enzyme you're looking at and you can't seem to figure out what class it should be, it's probably a lyase. Okay? And adenylate cyclase, this AC here, converts ATP to cyclic AMP, and it's creating this cyclic compound here, this bond formation, while eliminating the diphosphate here. And no oxidation's taking place, no water is used. So this is an example of a lyase reaction. Okay, group transfer reactions. Proton transfers common, methyl transfer, acyl, glycosyl. So all those groups can be transferred, but probably the most common, the one we'll talk about the most, I'm sure, is phosphate transfer. Okay, and this is to one, in metabolism, it's to activate metabolites, but then also this semester we'll talk about signaling in regulation, and group transfers, transferring phosphates is a very important step in regulation and in signaling. Right, and so our example here would be where we have a glutamate and we would add a phosphate group to it. Okay, so if we write it as one step, we have glutamate change to glutamine. But in a two-step process, we have a group transfer. A phosphate group from ATP is added to our glutamate, which activates it. Okay, then the amine group can be substituted for the phosphate group. Okay. Right. And these group transfers oftentimes are through a sp3, a nucleophilic displacement. Okay, so phosphorus is sp3, and it is an electrophile in that case, and this reaction occurs via a SN2-like mechanism. Okay, so a nucleophile forms with a partial bond to the phosphate center, giving a 5-coordinate intermediate or transition state. Okay, and that's typically, if you look at the mechanism of these group transfers, you'll typically see that. That would be the transition state. This 5-coordinate phosphorus group would be the transition state. All right, ATP is frequently the donor of phosphate, and that because it is a very high energy molecule. as you probably know, that ATP is the way cells sort of store energy and transfer energy, right? They make ATP by very favorable reactions, and then they use ATP to drive unfavorable reactions, right? So in this case, we have glucose. This would be the nucleophile, the OH group on the end of glucose here would attack the terminal phosphate group of ATP. Okay, then you would create a 5-coordinate intermediate here, and then you'd get your electrons would move, right, would break, and you would transfer that phosphate group from ATP to glucose. Okay. The last class of enzymes we'll talk about, and actually class number 1 in the classification system, are the oxidation-reduction reactions. Oxidation-reduction, we sometimes refer to that as redox, okay? And what redox stands for is reduction, oxidation. And that's characterized by a gain or a loss of electrons. Okay, oxidation, you can think of that as combining with oxygen, but more typically in metabolism, you don't really see that combining with oxygen all the time in every redox reaction. More typically, it's the loss of electrons. Okay. Reduction is either losing, it can be classified as losing an oxygen or gaining hydrogen, but really the definition for reduction is gaining electrons. Okay. And so to remember it, I always remember that reduction means gaining. Okay, those words are sort of opposite there, but reduction means gaining electrons, and oxidation is losing electrons. Okay, that's kind of how I remember it, but the reason why they're reduction, they're labeled oxidation and reduction, is it's because of the oxidation number. Okay, if you looked at the atom and the molecule that this redox is occurring at, in the case of oxidation, the oxidation number would be increasing, and in the case of reduction, the oxidation number would be becoming lower. Okay. So in metabolism, we use organic compounds as our fuels, and electrons can be stripped off of these during oxidation, okay, during metabolism, okay, and those electrons then are eventually fed into the electron transport chain, which pumps hydrogen and creates a hydrogen gradient in the mitochondria, and that's used to form ATP. Okay, not so important right now in our class that you know that. Okay, that's in metabolism. But here's an example of the fuels, and the things that are most reduced are hydrocarbons that are alkanes. Okay, then if they're being oxidized, it might turn into an alcohol. Oxidized further, we'd get a ketone, carboxylic acid, and then the most oxidized thing, at least for aerobic organisms, would be carbon dioxide. That carbon is in the most oxidized form. Okay, and again, like going through this, it's not very obvious that they're losing electrons. Okay, but we'll talk about that more in metabolism. Okay. Typically, when you get I should mention too that when you have a loss of electrons from something, or a gain of electrons by something, those electrons had to be lost by something else. So you never get an oxidation without a reduction of something else. Okay. So electrons are typically transferred, but then also the other thing that is transferred with the electrons are protons. Okay, so you get And typically how this occurs in dehydrogenases, which would be the class of oxidoreductases, class 1 enzymes. It's going to proceed via stepwise transfer. First, a proton is transferred, and then a hydride. Okay, and the hydride is a hydrogen atom that has 2 electrons. Okay, so that's a 2-electron transfer. Right, and in this case, we're lactate going to pyruvate. Right, we're transferring protons, 2 protons and 2 electrons. Right, and then those electrons, via cofactors that are being reduced, get fed into the electron transport chain. Right, and some common redox cofactors, NAD and NADP. Okay, NAD stands for nicotinamide adenine dinucleotide. Okay, and then the P at the end of NADP it's the same thing. There's just a phosphate group. So here at that hydroxyl group, at this hydroxyl group of NAD, NADP would have a phosphate group there. That's the only difference. And if you look at this molecule, it's rather big, complex. Here's the adenine dinucleotide, and this is the nicotinamide. Okay, this is the main important part of this molecule and what it does its job. So all this part here, we have a adenine diphosphate, ADP, and then another ribose sugar, right? All of that is just, really doesn't do anything in this molecule. This nicotinamide is what is taking the electrons and taking the protons. Okay, so when that's its oxidized form, there's a plus charge there. In the reduced form, there's an extra hydrogen here, and this nitrogen has the lone pair on it. Okay. Right, so that's all this is doing, is taking electrons. It's used as an electron acceptor. The oxidation state of NAD and NADP, when they're oxidized, they're called NAD plus and NADP plus. When they're reduced, we call them NADH and NADPH. But the different oxidation states can actually be tracked via UV spectroscopy. And you can follow this introduction here of a peak at 340 nanometers, and that tells you that it's reduced. So if you follow that, watch that grow in, you can actually watch and quantitate the electron transfer in these reactions. Okay. There's also flavin cofactors. Okay, and flavin cofactors are even a little bit more complicated here. This is the flavin mononucleotide, FMN, is shown here. You have this three ring structure. That's where the electron transfers occur. There's a ribose sugar, 2 phosphate, so 1 phosphate group, that would be flavin mononucleotide. Flavin dinucleotide has another phosphate group, and then basically an an AMP added to a flavin mononucleotide. So flavin adenine dinucleotide FAD is this big structure, and flavin mononucleotide is this smaller one. Okay, but they both do the same thing. They accept electrons. Flavin is different because it can do one electron transfer at a time, so it can accept one electron, and it actually has a free radical there when it does, and that's what's known as the semiquinone form, and then it can accept another one and be fully reduced. And this one electron transfer, one electron at a time, is very important because it allows it to interact with molecular oxygen, and it allows molecular oxygen to be used as the ultimate electron acceptor, which it is in aerobic organisms. Okay, we've seen a couple of examples of these coenzymes or cofactors, and I think it's important to define that. Okay, what coenzymes or cofactors are organic molecules that are required by certain types of enzymes to carry out their catalysis. Oftentimes they are vitamins or made from vitamins. Okay. Some examples, we've seen NAD, NADP in the flavins. Some other examples are things like thiamine pyrophosphate, TPP, lipoic acid or lipoamide, FAD, flavin adenine dinucleotide, and then also the NAD, and then coenzyme, something called coenzyme A. Okay. So if we just, for some definitions, so a prosthetic group or a cofactor is a coenzyme or a metal ion that is very tightly or covalently bound to an enzyme or a protein. Okay, so a cofactor, when we use that word, technically it's something that is either covalently bound to a protein or very, very tightly so it doesn't come out. Okay. I guess the other term would be coenzyme, and that is something that can go into the enzyme and come out of the enzyme. Okay, so NAD is a classic example of something that is a coenzyme because NAD can bind to the enzyme and be released. And then the reduced NADHs are what then go to transfer electrons to the electron transport chain. So prosthetic group or cofactor stays in the enzyme. A coenzyme can go in and out of the enzyme. Okay. A holoenzyme is holoenzyme is the complete catalytically active enzyme with all the coenzymes and cofactors in it. Okay. An apoenzyme or an apoprotein is the protein part of the enzyme that doesn't have the cofactor or cofactors in it. Okay, so holo is the complete structure, apo is missing cofactors or coenzymes. Okay, list of some typical enzyme cofactors and the enzymes that they're in. Okay, you'll see, I wouldn't expect you to say to necessarily be able to match these up, or if I give you a coenzyme, name the enzyme it's in unless we're specifically look at some of these examples, but they're shown here in the table, and as you go on in biochemistry, you'll see these many examples of enzymes that use these. Okay, TPP, FAD, NAD, pyridoxal phosphate, coenzyme A, biotin. Okay, those are very common. Okay, metals are also common as cofactors. Zinc, magnesium, nickel are probably the most common, I would say. But then there's even molybdenum, selenium, manganese, and also even potassium. Okay, So metals can be used as cofactors. Okay. If we looked at a specific example, and this is the conversion of pyruvate to acetyl-CoA by the pyruvate dehydrogenase complex, this is a good example because it has so many of these cofactors and coenzymes in it. It actually requires 5. Okay, TPP, lipoate, lipoic acid, and FAD are prosthetic groups, and then NAD and coenzyme A are co-substrates or coenzymes. Okay, so what this enzyme, and this is a very complex enzyme. It has 3 different parts to it, what are known as E1, E2, and E3. But then it's not just like one of each of those units in this complex. There's, I think, the E2. There's something like 60 of those, and I don't remember the numbers exactly. But there's, many, many, many copies of each of these parts that make up this complex. So the complex is huge. Okay. Right. So what it does is it takes pyruvate and it chops off a CO2 of pyruvate, and it attaches what's left of that acetyl group to coenzyme A. Right. Pretty simple reaction, but it uses 5 of these coenzymes and cofactors to accomplish this task. Okay, so it needs a coenzyme A to attach that acetyl group to. It needs an NAD to accept electrons, and then the TPP, lipoate, and FAD are involved in actually transferring everything. Okay, the mechanism shown here. This is the TPP group, but really all you need really the thing that's used in the TPP is this part. Again, not going to test you over this. Just an example. This is pyruvate. Okay, the acetyl group is attached to the TPP, right there. Okay, that acetyl group is then transferred from TPP to lipoic acid. Okay, that lipoic acid then can attach the acetyl group to coenzyme A. Okay. In doing that, this lipoic acid needs to be oxidized to accept that acetyl group. Okay, and this is the oxidized form of lipoic acid, and it's attached to a lysine residue in the enzyme. Okay. When it does it and it transfers it, it's now in the reduced form. So to convert it back into the oxidized form, that's when the E3 group of this enzyme, it uses an FAD. That FAD accepts electrons and hydrogen atoms from the reduced lipoic acid to form reduced FADH2 and oxidized lipoic acid. So it reoxidizes it. Okay, then that needs to be reoxidized, and that's done by transferring those electrons to NAD to make NADH. Okay, NADH then transfers the electrons to the main electron transport chain. Okay, so that's an example of, a mechanism you will see again if you go on in biochemistry. This is the pyruvate dehydrogenase complex. Whoops. Okay, catalytic mechanisms. So some of the basic mechanisms used by enzymes. Okay. Acid-base catalysis, covalent catalysis, metal-ion catalysis, and electrostatic catalysis. Okay, acid-base catalysis, you give or take protons. Covalent, you use covalent bonds with the enzyme and the substrate to change reaction paths. Metal-ion catalysis, you use these metal ions as either redox cofactors or as things that shift pKa values. And electrostatic catalysis, you get preferential interactions with the transition state and your positive or negative charges in your enzyme substrate binding site. In acid-base catalysis, okay, you can have protons transfer to or from water, okay, or you can have protons transfer to or from specific amino acids in the enzyme structure. So when water is used, we call that specific acid-base catalysis, and when residues are used in the enzyme, either basic or acidic residues, we call that general acid-base catalysis. So some amino acids that are used in general acid-base catalysis are shown here. Probably obvious: glutamate and aspartate, lysine and arginine. Right, so the acidic and the basic residues can be used. Maybe not so obvious, things like cysteine, histidine, serine, and tyrosine can also be used. So amino acids with OH groups, serine and tyrosine. Some that can be protonated and deprotonated, like histidine, and then also cysteine. Cysteine sort of looks like serine, right? But there's a sulfur instead of oxygen. Okay, those can all be used. Their acid form, where they'd be a proton donor, is shown here, and their base form, where they'd be the proton acceptor, is shown here. And if you count those up, that's like 8 of the 20 amino acids can be used in this catalysis. So actually quite a few. Right. This enzyme, chymotrypsin, is a peptidase. It breaks peptide bonds. Okay, and it uses general acid-base catalysis, and actually covalent catalysis too, to be honest. So the basic residue here can take a proton from serine, okay, and then that makes serine, the oxygen on serine, a better nucleophile. It can then attack the carbonyl carbon in the peptide bond of another peptide chain, and it forms a covalent bond there. It breaks the peptide bond, releases that half of that peptide chain, and then it has a covalent bond to this other part. Okay, so that's the first step in that covalent acid-base covalent catalysis. So chymotrypsin would be an example of both of those. Okay, metal ion catalysis, things like magnesium here, it can help catalyze reactions through charge shielding. So ATP has a lot of negative charges here on the 3 phosphate groups. If you have magnesium in your enzyme active site, that +2 on the magnesium, that +2 charge can shield some of the negative charges of ATP, okay, and help catalysis that way. All right. Lastly, we'll talk about some regulation of enzymes. Okay, there's 2 main types of regulation you can have. You can regulate the amount of enzyme that's made, or you can regulate the enzyme activity. Okay. So if you want to regulate the amount of enzyme that's made, you can do this at a few different levels, a couple different levels. One is the level of transcription. So we haven't really talked about that yet, but transcription is the process of taking DNA information and converting it into RNA. Okay? And then RNA is then converted into protein via translation. Okay, so you have transcription and translation to express genes into proteins. Okay, so you can either regulate the transcription level, which is how much RNA is made, or you can regulate the level of translation, which is how much protein is made from RNA. Okay, this tends to be slower, on the order of minutes. This regulation occurs. Okay, a quicker form of regulation actually comes when you you regulate the enzyme directly. Okay, after the protein is synthesized, you can have what's known as post-translational modifications. So post-translational means after the protein is made. Okay, this is a much more rapid process that occurs on the order of seconds or even less than seconds. Okay, this is probably a bit more complete description. 5 ways by which enzymes can be regulated by the cell. Okay, enzyme production, as we talked about, transcription and translation. Okay, you can also have degradation. That enzyme might be transcribed and translated into the active enzyme, but then it's quickly degraded. Okay, you can also have compartmentalization, okay? And that's the sequestering of the enzyme into different parts of the cell that are either, it's going to be used in that part, or it's just put aside in that part and not active. Okay. Activation and inhibition, those are the ones when we talk about signaling pathways, we're going to talk a lot about those. Okay. This is, you're either activating it or inhibiting it by molecules that, and typically these occur, they're molecules that are downstream in the process, in the pathways, and they feed back. And most typically when you have something like that, it feeds back and inhibits one of the steps of the reaction. Okay. Post-translational modifications, these are group transfer reactions, typically phosphate groups, but also methyl groups, and sometimes sugars are added to proteins to either turn them on or turn them off. And then finally, localization to different environments, which we really won't talk about that one. I don't think we'll see many examples of that in this class. Okay. So regulation can be through non-covalent modification, covalent modification. It can be irreversible and it can be reversible. Okay, some reversible covalent modifications are shown here. These are probably the ones that we'll see the most examples of. Okay, again, adding phosphate groups is one of the biggest examples of this regulation. Phosphate groups can be added to tyrosine, serine, threonine, and histidine. Okay, so 4 amino acids that phosphates can be added to, either to activate an enzyme or inactivate it. That, I would say, is the number one thing that we'll see in terms of regulation and in terms of signaling pathways. Okay, but some other examples are shown here. Okay. This example, it shows you just how complex this regulation can be. What we're seeing here is the interaction of enzymes in a pathway that deal with glucose. Okay, and in specifically in relation to insulin and glucagon. Okay, this pathway, you're not going to need to know it right now, but you'll see this again in metabolism. Okay, and what you see here is with the green triangles, those are examples when you have an increase or an activation, and the red X's are when you have an inactivation. Okay, so this enzyme, phosphorylase kinase, is active when it's phosphorylated. Okay, and the hormone or signaling molecule glucagon first increases cyclic AMP, and then that induces a protein, activates a protein called pKa, which takes ATP, a phosphate from ATP, and sticks it to phosphorylase kinase, okay, which is the active form. Okay, the other enzyme, or excuse me, signaling molecule, hormone, insulin, activates an enzyme called PP1 that uses water, hydroxylate, hydrolysis, there we go, reaction that cuts off the phosphate. Okay, so it removes phosphate from phosphorylase kinase and inactivates it. Okay, so that's a little bit of a preview of some of the regulation we'll see in when we talk about signaling pathways later on in this semester. Okay, but then you'll notice there's another step to it, right? We have this enzyme phosphorylase B active when it's phosphorylated. Okay, phosphorylase kinase, when it's active, will phosphorylate phosphorylase B to phosphorylase A and activate it. Okay, and then there's certain molecules here that inhibit this enzyme or inhibit this enzyme. Okay, and those are typically things like ATP. Right, if you have enough ATP, you don't want this to happen. Okay, AMP, on the other hand, if you have a lot of AMP around, that tells you that the cell maybe needs more ATP, so then you want this to be activated. Okay. But there's many more steps. So this is very complex regulation, which again, this is for this point in the class, don't necessarily need to know all these details. We're going to see this again when we talk about signaling pathways. Right. And then the last example the book gives is this enzyme ATCase, right, which the full name shown here. Okay. What it does is it takes carbamyl phosphate and aspartic acid, aspartate, and it attaches them together. Okay. This is an example of feedback inhibition, right? Because this is the first step in the formation of the nucleotide CTP. Okay, so if you have a lot of CTP around, what that does, the high CTP concentration will feed back and inhibit. Right, this is multi-steps to get here, but if you have a lot of this end product, it feeds back and it actually inhibits that enzyme ATCase, which is the first step of this process, right? So if you have enough CTP in the cell, it's going to inhibit that first step of the enzyme, so you're not wasting any of your starting material. Okay. In ATCase, it's actually a very good example, very similar to hemoglobin in that it has the T and R states, right? A tensed and relaxed state. Okay. So when we have nothing bound to this ATCase, it's in its tense state. The binding of your substrates, the carbamyl phosphate or aspartate, changes the shape of this enzyme. It rotates into the R state. And a better picture of that shown here. So in the tense state, okay, you don't really have a lot of room there between your subunits. Okay, when it rotates, it's a more open or relaxed structure. Okay. CTP preferentially binds in the T state, while ATP preferentially binds in the R state. Okay, so it has some actual activity with ATP as well. Okay, the curves, much like hemoglobin, when we looked at hemoglobin with this allosteric the allosteric effects in hemoglobin, we also see a little bit of that here in ATCase. Okay, the T state curve if we're looking at the concentration of aspartate as that increases and the rate of your product formation. Okay. In this T state, it kind of slow growing, and in the R state, it grows really fast. And what you get is this kind of composite in reality, because it starts in the T state, you bind, and then it converts to the R state, and then you get your quick product formation. Right, so that's all we have for the first part of enzymes. The next lecture will deal with the second half of chapter 6.